first of all, thank you so much for giving me the opportunity to give this talk today. I'm kind of excited about where the research is going and thought it'd be a good time to sort of, share what my research is all about. So today I'll be talking about sugar and cancer. So when I say sugar, you might think of what you see on the left most likely. But that is not the type of sugar that I'll be talking about today, I'm afraid. So that if you're a glycobiologist in the audience, you would know what I'm talking about. So today I'll be talking about sugars in the context of coating present on your cell. So until recently, even I didn't know that all of our cells have sugar coating on them. And these sugars are basically made up of 7 different monomer units, 7 to 9 different monomer units. Like you can see here, they could be glucose, mannose, fucose, galactose, sialic acids, and the likes. And they are basically these complex structures of multiple monosaccharide or multiple of sugar units brought together to form complex structures. And these have major biological roles. So initially in the 80s, when they were just about being discovered, people thought that they just were used as a structural component to provide like a protective layer for the cell, to protect it from its surroundings. But now we know that it is used for a lot more biological functions. So it is primarily used for cell-to-cell communication. So these sugars are a way of communication between different types of cells. And this communication in turn leads to immune responses, which in itself is a major function for these sugar molecules. So they coordinate immune responses. And just because of this one fact, I can like safely say that these sugar molecules are affected in every single disease that affects humans. So any disease has significantly altered sugar structures present on their cells. And these sugar structures are dynamic. So you might have heard of a genome, right? That's like all of the genes in a cell is called a genome. So all of the carbohydrates or all the sugars present on the cell are called the glycome. And these glycomes are constantly changing, they're dynamic. So as a cell changes, so does the glycome, which is why they are valuable markers for various disease conditions. And I guess in this day and age, I guess it's time. I guess you kind of have to talk about COVID. And interesting enough, there are really, interesting connections between these sugar coatings present on the SARS-CoV-2 virus that causes COVID. So interestingly, it was recently found that the spike protein that we all now very well know that binds to the ACE2 receptor on our lung cells is completely coated with these sugar molecules that you see in blue and green here on the left. And this is a molecular dynamics simulation of how much of these sugars are present on these spike proteins. So the main mechanism by which they aid the virus is that they mimic a human cell. So as I said, all of our human cells have these sugar coating present on them. And the virus is trying to mimic that by having the sugar coating on them, because of which our immune cells are not able to detect them. And that eventually leads to the virus successfully going and attaching itself onto the lung cells, and then hence causing a successful invasion for the virus. So yeah, just moving forward, just know that sugars are important for any disease to occur, and it is important to study these along with the vast amount of studies that we are currently doing for genomes, say sequencing or proteomic analysis. So glycoproteomic analysis, I believe, is just as important to be carried out to have a better understanding. Coming back to the context of cancer, which is what my PhD is about. So what essentially cancer is it's a communication breakdown. And as I've just stated, sugars play a major role in communication. So if we put these two things together, we can hypothesize. I mean, a lot of researchers have previously hypothesized that these sugar structures on cancer cells are likely to be different from normal cells. And this has been established quite a lot now. And you can see that these structures, these building blocks, look different in normal cells than they do in cancer cells. And because of these differences in these sugar structures, there are several consequences to this. So this leads to the development of cancer in itself. So it leads to the early development of cancer. So as a normal cell transition towards cancer, you can sort of see these glycoproteomic changes happening. You can also see that these cancer cells are now able to evade immune response. So an immune cell cannot distinguish between a cancer cell and normal cell because of these modified sugars present on their cell surface. And finally, it has also been proved that these alterations in sugars lead to progression of cancer from its earlier stages to late stages, and then eventually to metastasis, where it spreads to other parts of the body. But I'm more concerned about early detection, and so is this entire program that I'm a part of. This kind of graph, I guess, highlights why this is so important. It's primarily because survival is just much higher when the diagnosis happens at the earlier stages, which is stage 1 and stage 2, which is intuitive, which we have previously known. But it's baffling still to me to sort of note that the survival is more than 3 times higher when cancer is diagnosed early. And this is just using therapeutic approaches that we currently have. So we don't need any more fancy targeted therapeutics than we already have, although that would be really useful. But just the technological advancements in terms of therapeutics that we already have would be enough to save 3 times more people if we have the diagnostics technologies on par with the therapeutics. So coming in context, bring it all into the context of esophageal adenocarcinoma, which is the particular type of cancer that our lab focuses on. This is a cancer that has increased 7-fold in the UK in the last 30 years. It is one of those only cancers that is increasing in trend when compared to the other major cancers like lung and breast cancer, at least at that unprecedented rate. And there is the survival is one of the worst, because it's about 13% for 5-year survival, which is to say that only 13% of patients who have been diagnosed as patients with esophageal adenocarcinoma would survive beyond 5 years. And finally, the more promising aspect of this statistic is that if detected at the precancerous stage, which is the earliest stage, the survival jumps up to greater than 80%, which is what our lab is I guess, focused on delivering. And on the right, you can see the progression that of a normal tissue that leads to adenocarcinoma in the end, which is the cancer. So the first transition that occurs is this thing called the Barrett's esophagus. So what Barrett's is basically, you might have had, or you might have known people who have heartburn symptoms, right? So heartburn eventually leads to, is basically caused due to these reflux symptoms that happen, where contents of the gastric cardia, contents of the stomach is sort of refluxed back into the esophagus. And this refluxate is like highly acidic in environment, right? So if that happens over a period of time, that eventually leads to transformation of cells in the esophagus, to this, from a normal squamous type cell to a more like this globule type of cells, and we call them goblet cells. And that's I guess, the characteristics of Barrett's esophagus. And as our lab focuses on early detection, as I've said, we are focused on trying to detect Barrett's esophagus, which is the primary transition that eventually leads to cancer. but there are several diagnostic challenges of esophageal adenocarcinoma, which is why the survival rate is quite low at the moment. So it is asymptomatic until late stages. So the symptoms are very, very general. There are no specific symptoms that would lead a clinician to refer a patient to get a more invasive test, such as the endoscopy, which, as you can see, is quite unpleasant, where a scope is sort of plunged down your throat and some biopsies are taken and then imaged basically. And this is a major barrier for both patients and healthcare providers, because for patients it's an invasive process, and for the healthcare providers it's an expensive sort of test. So not everyone in the population can undergo this sort of testing. Which opens up a clinical need for a new objective biomarker test. So by a biomarker, I mean like any sort of biomolecule, like I said, the sugars in our case, that could be used to stratify high-risk patients. So that we can pick out patients at high risk from a general population who can then undergo endoscopy, so that you don't need to, you don't need to subject the entire population to this invasive test. And also it should be low cost and easy to administer, so that people can actually adopt it quicker. And so this is what our lab had previously worked on, which is this technology called the Cytosponge. So I'll try to play this video. Hopefully it works. So you can see that a patient has swallowed a capsule at this point in the video. And the capsule sort of goes down the esophagus into the stomach, as you can see. And the capsule contains a sponge, which eventually is opened up as the capsule dissolves. And that takes about like a minute or so, I think. About like 5 minutes in total, actually, for the capsule to completely dissolve. And then the nurse is basically just pulling it out. And I've done this, it's not as unpleasant as it looks, but as it's pulling it, as the nurse is pulling it out, you scrape these cells along the length of the esophagus. And as you do that, you pick out these cells that are super important to us, because you're getting a localized sampling of the esophagus, which is then put into a preservative liquid, and then it's shipped off to a centralized lab facility, where it is then, as you can see on the images on the right, it is then put onto glass slides, and then a pathologist looks at it under a microscope to see these types of cells. And if they see these these globule types of cells under the microscope, then it is to say that, yeah, the patient is at risk of potentially developing esophageal adenocarcinoma, because the patient is positive for Barrett's esophagus. But this whole process takes weeks, and it's it's a very lab intensive and skill intensive process. And the objective of my PhD, I guess, is to to limit the workflow needed to get a result, as opposed to requiring a lot of lab procedures and a lot of burden on pathologists to do the testing. So as I've spoken about sugars already, so glycan sugars could be a great biomarker to be employed for samplings that you get from the Cytosponge. That's how the PhD began, and we're still working towards characterizing different biomarkers that can pick out these sugars initially, and then hopefully build a test around that, so that you don't need a very lab intensive process that takes a long time to give you a result and costs quite a lot of money. So what we started off doing was we needed to get a proof of concept initially that these glycan sugars are actually in fact altered in Barrett's esophagus, because that hadn't been done before. So it was, although several cancers like breast cancer, colorectal cancer has been vastly studied for glycoproteomic analysis and sugar structures, esophageal adenocarcinoma is quite poorly understood in terms of its glycosylation landscape. So because of that, we had to identify some markers first, and our approach of trying to identify these glycan sugar markers was using this protein known as lectin. So a lectin is basically just if like for biologists in the audience, they'd know what if an antibody is, which just binds to another protein. But in this case, a lectin is also a protein, but it binds to sugars. So it's just a protein that binds to sugars. That's all you need to know. And these lectins are cheap. So they are 1/10 the price of an antibody. So they're quite easily sourced. In fact, I get my lectin sourced from a coral tree sometimes, and then sometimes from a peanut, and sometimes from a shrub and stuff like that. So it's quite easily sourced compared to a monoclonal antibody, which needs to be produced specifically under lab conditions, which cost a lot of money. So ideally, this seemed like a good way of trying to detect these sugar markers, because eventually our goal is to develop a test around it. So having a cheap way to detect these markers would be ideal. And the way we went about doing the biomarker discovery analysis was to basically buy these lectins that are tagged with a fluorophore. That is just a fluorescent tag that will glow green wherever it binds onto a tissue. So what I call now the Bell study, or in which is basically the Barrett's esophagus lectin staining study, is what. So how that began is to try and identify these biomarkers and try and validate whether or not lectins can actually be used to pick out these sugar-based biomarkers. So we got biopsy tissues from 5 different patients who were all confirmed Barrett's esophagus by the surgeon. And we collected 3 different sites in the body as the biopsy tissue. So we got the normal squamous esophagus, which is just the normal sampling. We got the actual disease area, which is the Barrett's esophagus tissue. And we also got the gastric body, or the gastric tissue. That is a confounder, or that is used as a reference, control reference tissue, especially for the cytosponge, because the sponge will go into the stomach. So we need that as a reference tissue as well. And also the normal squamous tissue is also a normal control. So we got 5 patients with 3 different tissue types for each patient. And we tested 20 different lectins using this immunofluorescence approach. And this is just a summary of different lectins that we tested. And we found that they are tissue specific, as we had hoped. And different lectins bound to all 3 different tissue types. So NE is normal esophagus, GC is gastric, which is the stomach, and BE is Barrett's, which is the disease condition. So we found that 5 of the lectins bound to all of these tissue types. So just to quickly mention, the lectins aren't actually named L1 through 20. I just had to change the labeling because of intellectual property concerns at this stage. So the first 5 lectins stain all 3 tissue types, and there were some lectins that stained for the stomach and, interestingly, the diseased condition. And we got one lectin that nicely stained goblet cells, which is exactly what we were hoping for. And also we got some other lectins that stained only normal tissues as well, which could be potentially useful later on. So we got 3 lectins that we could potentially use in a much larger patient cohort. And these are just some of our staining results. This was L6 that I'm calling, and this is the one that worked best for us. So there's what you can see here is that there are 3 tissue samplings. So the first one is a normal squamous esophagus. So that's the normal tissue. So ideally, a biomarker for a disease shouldn't be positive for a normal tissue. So that is good that nothing is binding there. For the gastric cardia, we do see quite a lot of intense staining in the epithelial surface, which is also good because eventually we want to use this for the cytosponge, and the sponge enters the stomach. So we do expect these cells to be present. So it's nice that it is positive here. And it is also positive, very interestingly, for the Barrett's tissue, which is what exactly we were looking for. And as you can see here on as marked by the red arrows, they are these globule type of looking cells, which are present on the surface of the epithelia, as you can see under a normal H&E, which is what a normal pathologist would use to make this sort of diagnosis. And they are present at the same location, which kind of validates that, yeah, they are in fact goblet cells that are being picked up. So we did have other lectin stainings, as I've previously stated. So we did get some lectins that stained only the gastric cells. So they did not stain Barrett's tissue at all, and neither did they stain normal tissue, but they only stained gastric cardia, which you can see in green intensely here. So this could potentially be useful as well in conjunction with this lectin, because this one stains both markers, which is the stomach and the Barrett's. And in this case, it's only staining the stomach or the gastric tissue. So we could potentially look towards a subtraction, a signal subtraction sort of a methodology where we use 2 or 3 lectins in conjunction to try and get the signal that that we get from Barrett's tissue only. And that's basically what this heat map is presenting as well. So this is just a patient summary of lectin L6 and lectin L12. L6 is the lectin that I showed that bound to goblet cells in the Barrett's tissue, which is the diseased tissue, and it also bound to the gastric cardia, as indicated in purple. And the lectin L12 bound only to the gastric cardia tissue or the stomach tissue, and did not bind at all to the Barrett's tissue. So eventually we could look at subtracting lectin L12 from lectin L6 to get a disease signal only, and that is one approach that we're considering going forward. So in addition to the biopsy sampling, we also did a proof of concept analysis just to make sure that the sponge samples can also be used for this lectin based analysis of sugars. So interestingly enough, the same results as we saw in the biopsies was replicated. So this is a bit dirtier, as you can see from the biopsies, because it's not a clean cut. I mean, it is a sponge that we collect cells from and then just place it on a slide. So it's not going to be as clean, but then again, you can see that it is negative for normal squamous tissue. So it is not binding any of the normal, and it is only staining columnar tissue, which is the gastric tissue. And we can also see some of these goblet cells being picked up again. So that is again so that's the same as what we found in biopsies, is also being replicated in the cytosponge samples. So that goes to show that we could use these lectin markers to query sugar-based biomarkers from the cytosponge to detect Barrett's esophagus, which is the earliest stage of esophageal adenocarcinoma. So now that we have this marker, I guess the next step is to try and build a test around it, because the way we're currently doing this is, again, we're taking tissue sampling, again, shipping it off to a centralized lab facility, doing immunofluorescence staining in the lab, which takes hours, and we need to let it develop over a day, and then use highly expensive fluorescence scanners to take these images. So that's not an ideal point of care test, right? So that's what you would want with a test like Cytosponge, so that you can quickly get an answer within the GP's clinic, for example, where you would do the test and then eventually get an answer within the hour. So in working towards that, I sort of, I guess the way to do that would be to build a biosensor. So what a biosensor is essentially, it's defined as an analytical device that converts a biological response into a detectable signal. So that's the textbook definition. But essentially, to me, what a biosensor is, or an ideal biosensor is this Star Trek tricorder. So anyone who's seen Star Trek would know that, so this is like an amazing piece of futuristic technology that we all would love to have. And what this enables is personalized medicine. It couldn't get more personalized in that, I guess. And I guess what it means is it also enables point of care diagnostics. So by point of care diagnostics, I just mean that the test comes to you rather than you traveling all the way to a centralized lab facility or to a hospital setting to donate your sample, which in turn goes to another lab facility where it is processed, and then again the results are shipped off to you. So instead of all of that happening, this is a more decentralized approach, I guess, where the test comes to you, and then it's much easier for you as the patient to be tested as well. And that is the future, I guess, but we're not there yet. So this is what a current biosensor would look like. And so this is a little bit of a plug. So we were at Imperial before coming to Cambridge. We were part of this. Our lab published this book on bioengineering innovative solutions for cancer, and that talks about an entire chapter on how to build a biosensor. But a quick primer is just that it involves 3 different modules. It's like a Lego brick construction where there are 3 modules that come together to perform one function. And so the first module is, of course, the sensing. So there are different things that we could potentially use to capture. So that's the capture element. So in our case, we're using lectins to capture sugars. So sugars is our analyte of detection, and lectins are our recognition elements. So that's what we're going to be using. You could use anything else like aptamers or antibodies to pick out whatever protein-based or DNA-based marker that you want to look at. And that would eventually, so the binding event of your marker to your capture element would eventually lead to different types of signal. So that could be detectable. So it could either be fluorescence, like I previously shown, or it could be colorimetric, so that you could like visually see that like the best example is a pregnancy test, where you can see that the test turns purple or pink or whatever. And because of that, you can sort of tell whether it's positive or negative, which is ideal for a yes, no sort of an answer wherever you need it. And electrochemical. So that's the more complex one, I guess, where you get quantitative numbers, like in the case of a glucose sensor, where you get a number. So all of those glucose sensors are electrochemical sensor, wherein a binding event in this case, in that case would be glucose, would lead to an electrical signal that is then being converted to a particular number of glucose concentration. And then that would, you'll need to process that signal to eventually give you a concentration as to how much of this marker is present, or in our case, how much of the sugar is present in the solution, or in our case, it would be the sample that you collect from the sponge. And several different materials have been used so far. People have used glass, people have used silicon chips, people have used polymers, several different types of polymers, like PDMS is one of them as well. And interestingly enough, people have used just plain paper to do these types of sensing as well. And this is just to give you an example of what a working sensor would potentially look like. This is an example of an optical biosensor. I'll come to electrical soon. This was the work I was involved in previously, where like I said, this is like a lateral flow assay, it is called, where it's like a pregnancy test assay, where you load your sampling on a sample pad that eventually goes to a test line, and your capture probes would pick out whatever you're targeting. In our case, this was just a blood sample. And we picked out targets, what are our targets of interest, and then we could get a clear signal that you can see here, like a test line, like a dark line. And that line we eventually quantified, and we got this nice graph. So these 3 plots are all you need to essentially prove that your sensor is working and it could be used in a clinical setting potentially. So this is called a proof of concept study, right? So so the first plot is sensitivity. So the more amount of your target sample you add, the more signal you get. That is intuitive. So that if that goes up with increasing concentration, you know that your sensor is working. So, and you need to know how low you can go. That's called the limit of detection. And you also need to know something known as a dynamic range. So that's the range in which you get a linear response. And we got good sensitivity with ours. And you also need to prove specificity. So you need to try and add other target analytes and show that you don't get a signal, basically. And you only get a signal when you add your target molecule. And that's what we showed. We were trying to try and target a 150, miRNA 150. And we targeted that, we got a high response. And then whenever we incorporated a mutation in that DNA, our signal dropped quite drastically. So that is another key uh, piece of test that you will need to do to prove that your sensor is working. And finally, you need to do some sort of clinical testing. So we had around 20 clinical patient samples where we were trying to predict with this marker, we were trying to predict women who would eventually go on to deliver preterm in this case. So we found that this marker, and with using this test, we could predict patients who eventually delivered preterm because of we got statistically significant differences between at term delivered and preterm delivered patients. And we got blood samples from 20 different patients to do this type of analysis. So that's everything you need to show that your sensor is working. Now that we know that, so coming back to electronic sensing, which is what I'm doing now currently. So in terms of electrochemical sensors, there are lots in the market. You might have heard of like several different types of electrodes, metal-based electrodes, gold, platinum, silver. They've all been used as electrodes. But the next frontier, I believe, are this material known as conducting polymers, especially for biosensing applications. These conducting polymers are a great new promising class of electronic materials. And several different ones have been identified till date, but the ones we used most commonly is called PDOT, with this long chemical name here. But this PDOT has been shown to be the most electrochemically stable amongst all of them. So it's the most widely used for biological applications at the moment. And these conducting polymers act as very efficient electron relays between a biological molecule and the transducer. So it was, if you remember from the primer, from the how to build a sensor, it's just the first part and the second part. So it acts as a good relay between your whatever you're attaching as a recognition element. In our case, it would be the lectin, and with the transducer element, which would be the electrode below it. And people have done several different types of studies using this type of conducting polymers. They've done breath-based sensors for detecting alcohol, and then there's been saliva-based sensing. There's also tear sensing using like embedded sensors and contact lenses. And Google has sort of gotten on that as well at this point. And there's, of course, several other types. So interstitial fluid, which uses some microneedles, sweat-based and implantable. So these are all wearable devices that have been used, that have used conducting polymers. So these are a great class of novel materials that we could use to do some electrochemical sensing. And our approach is just it doesn't require any sort of flexibility or variable aspect to it. It's just a simple in vitro assay that where we're gonna take some cells from the sponge sample and then just pipette it onto a chip, which would contain these polymer electrodes. And using that approach, I've initially, a while ago, we got these plots that kind of point towards the direction that we have a working sensor. So in this case, we used PDOT, as I said, which is the most stable conducting polymer. And so in all of these plots, the black curve represents the plain PDOT, which is just the polymer. And the green curve here in all these cases represent lectin L6. If you remember, that was the marker that worked best for us. After lectin, we added the sugar, S6, which is specific to that lectin. So hopefully we would get some specific binding in that case. As you can see, this is sort of, I won't go into the depth of what these curves mean, but it's intuitive to see that your signal kind of goes up at every level of addition of a molecule. So with plain PDOT, you can see that the curve is quite low here. And then you get a much nicer semicircular response when you add your lectin on top of this electrode surface. And you get an even bigger signal when you actually load your sugar, which would then bind to your lectins that have been previously coated on the electrode surface, so that you get a much bigger signal. And three different plots are classically used to make these sort of analysis. So it's the Nyquist plot, the Bode plot, uh, which is also used in conjunction. So where you kind of query high frequency ranges and low frequency ranges to try and estimate what is actually happening on your electrode surface. And in this case, you can kind of see that the plane P dot gives you a tiny peak at the high frequency range. And then there's a shift occurring there that leads to when you add your lectin, and then that eventually goes even higher when you add your sugar. And this kind of is seen in another cyclic voltammetry plot, which is another way of getting similar data as well. So they're all used in parallel. So it's not that one is preferred over the other. So they're all complementary to each other, trying to give you similar kind of understanding of what's happening on your electrode surface. So that is basically what we're currently working on. So once we have this sensor, we would then eventually work to put it on, query cellular sampling on this, and then try and test actual clinical samples from the Cytosponge, and then try and get similar responses where we could detect the sugars. So this is what we're trying to do. So in summary, we would just get the sponge samples with specific glycan sugar markers. We would put them onto somewhat of a microfluidic chip. But in this case, for those of you who have worked with microfluidics would know that you would require complex pumps and a lot of lab skills to sort of generate a working microfluidic. But in this case, there have been previous reports from our lab where you could just press the button and get some fluidics to move through different electrodes that are functionalized with different recognition elements. And the goal is to incorporate nanomaterial on these electrode surfaces as well, so that would drastically enhance the electrochemical signal as well. And we are querying different types of nanomaterials to get different types of responses. And the goal is to detect these glycan sugar molecules from our clinical samples. And hopefully towards the end of my PhD, I guess the goal would be to get a similar kind of plot where you can estimate the degree of risk of a patient having Barrett's esophagus by getting these types of curves, where like a small curve would represent low risk of a patient having Barrett's esophagus, and medium risk and high risk. And high risk would eventually, hopefully we'd have enough clinical evidence to show that it requires at some point of intervention or further testing at that point. So that would be the ideal case scenario. So that's everything I had to present today. It's still ongoing work. And just to give you a quick recap of everything I've spoken about, because it covered a lot of different fields. So to highlight, sugars are important. They're a part of several different diseases. So getting a better understanding of these sugar coating on your cells is quite useful for a lot of different applications. And a sponge is less invasive than endoscopy. And I speak with experience because I've actually undergone the Cytosponge test, as you saw in the video, and it's not at all that unpleasant. It's just when at the point where the nurse is pulling it out, where it might be slightly unpleasant, but just for a second. And that's nothing compared to what a patient has to undergo for an endoscopy. So it's much less invasive. So it's much easier to administer. And biosensors enable precision diagnostics. So I guess better biosensors that are more reproducible need to be, I guess there's a need for those, because there have been multiple designs for biosensors over the last decade, but very few have actually translated to the clinic. And I believe the main reason is reproducibility, especially in case of electrochemical sensors. There's a huge issue of reproducibility, but hopefully using these conductive polymers and some nanomaterials to enhance the signal, hopefully we'd get around that issue of reproducibility. And yeah, so as I've said, so we would integrate nanomaterials to enhance sensitivity, because the amount of actual Barrett's cells, or cells that have these disease-like appearance, are very, very few. So we get about, I guess, about 1 million cells from the sponge, and only about 10% or even less is actually cells from the Barrett's esophagus. So our sensor needs to be incredibly sensitive for it to be able to pick out these cells, and incorporating several different types of nanomaterials would be the way to go about it. And yeah, so that's one type of nanomaterials that we're currently working on, which is a magnetic nanoparticle, which is really fun to synthesize, because yeah, you can quite nicely see them get attracted to a magnetic stand, that, as you can see there. So we're kind of querying that type of nanoparticles as well. And finally, I guess I'd like to end by saying that potentially, hopefully in the future, sugar biosensors coupled with a sponge could one day help detect cancer. And I'd like to end with that. Finally, yeah, just to thank everyone, because this is, yeah, I'm actually part of 3 different labs. So I'm based at the MRC Cancer Unit, but I work heavily under Roisin Owens' group, which is the Bioelectronic Systems Technology Group, or otherwise called the BEST group in CUB, which is Chemical Engineering and Biotechnology. And I also work with Professor Liliana Fruk, who heads the Bio-Nano-Engineering group at the Chemical Engineering Department. So, and of course, thanks to my funders, the Cambridge Trust and Trinity College. And yeah, again, thank you so much for giving me this opportunity to present today. And I'm happy to take any questions you might have.